and it was formed by a vascular cambium that could not form new fusiform initials. In the vascular cambia of seed plants, as the cambium is pushed outward by accumulation of new secondary xylem, its circumference becomes greater, but fusiform initials divide by radial walls, creating new fusiform initials. An old cambium has many more cells than a young cambium. In arborescent lycophytes, cambial cells could not undergo this division, therefore, an old cambium had no more cells than a young one. Older cells became stretched circumferentially until they were no longer functional, secondary growth then ceased. Figure 2115 Lycopodium cernuum is a common species that has extensive rhizomes, vertical chlorophyllous shoots, and sporangia clustered into cones. These shoots are leaning against and being supported by surrounding grasses and shrubs. Lycopodium obscurum, with sporangia clustered into cones at the tips of branches. And Lycopodium lucidulum is one of several lycopod species in which sporangia are distributed among the leaves rather than in strobili. Selaginellas can be distinguished from lycopodiums by a small flap of tissue, the ligule, on the upper surface of selaginella leaves. Although ligules are simple structures, and their adaptive advantages are unknown, they are ancient and can be used to distinguish early fossil selaginellas from fossil lycopods. Isoetes is a genus of about 60 species of small, unusual plants called quillworts, which grow in wet, muddy areas that occasionally become dry. Their body consists of a small corm-like stem that has roots attached below and leaves above. Isoetes is heterosporous like selaginella, and almost every leaf contains sporangia. Microphyls in this genus also have ligules. Weak cambial growth results in the production of additional cortex parenchyma to the exterior and a type of vascular tissue to the interior. The latter tissue is a mixture of tracheids, sieve elements, and parenchyma. Stylites plants are very similar to those of isoetes, and several botanists have suggested that the two species of stylites are really extreme forms of isoetes. The anatomy and morphology preserved in fossil lycophytes indicate many instances of convergent evolution with seed plants. In both lines, elaborate, efficient leaves, wood, bark, and roots evolved. Sporangia became separated from non-reproductive organs and were grouped together into strobili. In both lycophytes and seed plants, heterospory evolved, as did endosporial development of the megagametophyte. In seed plants, megaspores were retained in the megasporangium and evolved into seeds. This nearly happened in certain extinct lycophytes. We cannot tell from the fossils whether convergent evolution of specialized metabolisms also occurred, but isoetes does have CAM photosynthesis. It also has a unique means of obtaining its carbon dioxide, absorption by the roots from soil or mud. Its leaves have a thick cuticle but no stomata. We can only wonder what the metabolism of the extinct tree-like lycophytes was like. The extinction of so many of the lycophytes is unfortunate. They were complex, sophisticated plants that had many superb adaptations. They probably underwent interesting responses to changes in season, and they must have been able to resist numerous types of pathogenic bacteria, fungi, and insects. Those aspects of their biology will probably remain unknown to us forever. Figure 2116 Plants of Selaginella wildenoi are large and occur in moist habitats. Plants of S. Lepidophylla are also large, but occur in habitats that cycle between wet and severely dry. When dry, as shown here, the plant desiccates, curls up, and becomes dormant. The brown color is only on the underside of its leaves, the upper sides are green, even in this dry condition. If moistened with rain or dew, it quickly rehydrates, uncurls, and becomes active. Plants of S. wrightii tend to be small, compact, and easily mistaken for mosses. These are desert-adapted and grow on bare rock in full sun throughout arid regions of the western United States. Figure 2117 The megagametophyte of some selaginellas develops almost entirely within the megaspore wall. The spore cracks open, exposing archegonia and permitting fertilization by swimming sperm cells. Microgametophytes of Selaginellas develop within the microspore wall, then liberate motile sperms. Figure 21:18. Although isoetes is a small plant, its short basal corm-like stem has a small amount of secondary growth. The megaphyll line of evolution, Euphylophytes. Trimerophytes. Division Trimerophytophyta was proposed in 1968 for 3 genera of extinct plants, Trimerophyton, Psilophyton, and Pertica. Their fossils strongly resemble those of rhiniophytes, having terminal sporangia that dehisce laterally, homospory, dichotomous branching, and an end-arch vascular cylinder of tracheids. Trimerophytes, however, are considered a distinct advancement out of rhiniophytes because of several special features. Most important is the trend of overtopping. Trimerophytes had an unequal branching in which one stem was more vigorous. In later species, the inequality was so pronounced that main stems and lateral stems can be identified easily. Finally, Pertica displays pseudomonopodial branching, that is, a single main trunk rather than a series of dichotomies. The plants have small lateral branches, some fertile and bearing sporangia and others sterile and acting as leaves. Simultaneously, the positioning of branches became more regular and controlled. In rhiniophytes, the points of dichotomy were irregular and unpredictable, but in some species of trimerophytes, lateral branches were arranged in a regular spiral phyllotaxy. Other types of phyllotaxy that occurred were alternate, decussate, disticus, tetrasticus, and even whorled. Correlated with the evolution of a pseudomonopodial growth habit and the presence of numerous photosynthetic lateral branches was an increase in the vigor and robustness of the plants. Individuals of Pertica quadrifaria had stems 1.5 cm wide and about 1 m tall, whereas those of P. dalhousii are estimated to have been as much as 3 m tall, approximately the height of tall shrubs today. Although a dense stand of P. dalhousii would not be called a forest, there would have been vertical stratification of light, air movement, and humidity within the canopy. Trimerophytes became distinct from rhiniophytes during the Lower Devonian and existed until the Upper Devonian period and then came to an end not by going extinct but rather by evolving into the ancestors of ferns and seed plants. Figure 21:19. Pertica quadrifaria had one main trunk from which grew small branches. The smallest twigs still show dichotomous branching, but larger stems branched pseudomonopodially. This branch pattern also evolved separately in lycophytes, but here in the trimerophytes, it is the ancestor to the stem structure of seed plants. The globular structures are clusters of sporangia. Origin of megaphylls. At least 3 distinct types of homoplasic structures called leaves occur in plants: leaves on gametophytes of non-vascular plants, innations microphylls of zosterophyllophytes and lycophytes, and megaphylls, leaves that evolved from branch systems and are present in all seed plants, ferns, and equisetophytes. Megaphyll evolution is summarized by the telome theory. Imagine a plant like Pertica consisting of a main stem and smooth, cylindrical, dichotomously branching lateral stems. The ultimate twigs, those of the last dichotomy, are known as telomes. Now imagine that all subdivisions of a lateral branch become aligned in one plane and that parenchyma develops between telomes and even lower branches. This is not a leaf, but it has suitable characteristics to be the ancestor to leaves. Also, the leaves we see on trees and herbs of angiosperms are the result of about 300 million years of evolutionary refinement. If the branch system involved in this evolution produced sporangia, the resulting structure would not be just a leaf, but rather a sporophyll, a sporangium-bearing, leaf-like structure. For one reason or another, the plants with this organization outcompete those with microphylls, the lycophytes. This is not to say that the megaphyll type of organization itself was the main reason for their success, but today, megaphylous plants are by far the more common. We believe that all megaphylous plants form a monophyletic clade, now referred to as the euphylophytes. Manilophytes. Several current studies suggest that megaphyllous plants are united by three synapomorphies. Their roots have exarch xylem, they have megaphylls, and they have a 30 kilobase inversion in the large single-copy region of their plastid DNA. The euphylophytes as presented in Figure 21.8 contains two sister clades, the manilophytes and the woody plants. Many manilophytes are plants we know as ferns, others have been called fern allies. If we want to use the term ferns in the sense of a monophyletic, natural group, we must also refer to equisetophytes and xylitales as ferns. This sounds strange to many experienced botanists because horsetails differ from all other ferns in many respects. Remember that Figure 21.8 is a model, a hypothesis. It would not be surprising to some of us if within a few years, new data would indicate that equisetophytes form a clade sister to ferns, but not part of ferns themselves. In this book, Equisetum and its clade will be treated as sister to the ferns. Figure 21.20. The leaves of ferns, Equisetum, and seed plants are megaphylls that arose by evolutionary modification of stems. In the first step, one branch overtops the other, which remains smaller and lateral. Next, lateral branch systems restricted their branching to just one plane, they stopped producing three-dimensional branch systems. Finally, the spaces between close branches developed a thin sheet of chlorophyll-containing cells in a type of webbing. Equisetophytes. Equisetophytes have been classified as division Arthrophyta. They consist of several genera of extinct plants and one genus, Equisetum, with 15 extant species known as horsetails or scouring rushes. The living plants are all herbs without any secondary growth, and although certain species may attain a height of up to 10 m, they are usually less than 1 m tall. Their aerial stems have a characteristic jointed structure, with a whorl of fused leaves at the nodes. The leaves are small and have just a single trace of vascular tissue, but they are small megaphylls, not microphylls. If branches are present, they alternate with the leaves rather than being located in the leaf axils. The vertical, aerial stems arise from deep subterranean rhizomes. In harsh habitats, aerial shoots die during winter, but rhizomes persist and plants can spread vigorously. True roots are present, being produced at the rhizome's nodes. All species in this group are extinct. Figure 21.21. Equisetum plants are usually small, having vertical shoots that arise from subterranean, highly branched rhizomes. In some species such as E. hyemale, each shoot is both photosynthetic and reproductive. In other species, such as E. arvense, some shoots are vegetative only, and separate reproductive shoots are produced at a different time of year. This is E. giganteum, in which the plants grow exceptionally large. Botany and beyond. Box 21-1 Molecular Studies of the Evolution of Early Land Plants. The evolutionary relationships of early land plants are still not understood well. Plants like mosses, liverworts, rhinia, and lycopods are so simple that there are not many characters to compare. Furthermore, when two groups do resemble each other, we cannot always be certain whether the common characters are homologous or analogous. Analysis of the arrangement of genes in chloroplasts has been used to investigate which early land plants are related. The circular DNA from chloroplasts has been isolated from many plants, then DNA restriction enzymes were used to analyze the order in which genes occur. Currently, many groups have been studied, and it turns out that mosses, liverworts, and lycophytes have one arrangement whereas euphylophytes all have an alternative arrangement. From numerous studies on many types of plants, it had been concluded that chloroplast genomes are quite stable and this type of rearrangement is rare. It seems unlikely that this type of rearrangement could occur twice, producing similar gene arrangements in three distinct groups. Thus, the presence of the rearrangement in all euphylophytes is strong evidence that all constitute a monophyletic clade, that manilophytes, early woody plants, and modern seed plants all shared a common ancestor. Figure B21-1 The order of genes on the circular DNA of plastids of liverworts, mosses, and lycophytes, and of seed plants. The section shown in blue is reversed in euphylophytes. Internal structure is as distinctive as external morphology. Stems have a pith, and thus, these are siphonosteles, not protosteles as in all other plants mentioned so far. Protoxylem forms next to the pith, interior to the metaxylem, and thus, it is endarch, a rhiniophyte trimerophyte trait distinctly different from the exarch protoxylem of zosterophyllophytes. Vessels are rare outside the flowering plants, occurring only in Selaginella, several ferns, 3 genera of gymnosperms, and Equisetum. Stem elongation causes some cells to be stretched and torn, forming canals. The cortex of Equisetum is composed of large cells, the outer ones chlorophyllous. Air canals may be located in the outer cortex. The epidermis contains stomatal pores and guard cells, as well as such large amounts of silica that the stems are tough and rigid. Reproductive structures in Equisetum are specialized. Sporangia always occur in groups of 5 to 10 located on an umbrella-shaped sporangiophore. This has a short stalk and a flat, shield-shaped head from which the sporangia project, parallel to the sporangiophore stalk. Sporangiophores are always arranged in compact spirals forming a strobilus. Equisetum is homosporous, therefore, all plants have only one type of sporangiophore and strobilus, but in some species, the strobilus occurs at the tip of a green photosynthetic shoot, whereas in others it is borne on a special, colorless reproductive shoot. After spores are released, they germinate on moist soil and develop into small green gametophytes. They have no vascular tissue and no epidermis or stomata, the body is just a small mass of parenchyma. Gametophytes of Equisetum are either male or bisexual. Antheridia release numerous multiflagellate sperms that swim to the archegonia and eggs. The megagamete and zygote are never released, but rather are retained and nourished by the gametophyte. Figure 21-22. The stem anatomy of Equisetum is dominated by numerous canals. The center of the pith is torn apart, forming a central canal or pith canal, and around the remnants of the pith are carinal canals formed by breakdown of protoxylem. Just exterior to the carinal canals are metaxylem and phloem, vascular bundles are endarch. The cortex contains cortical canals that alternate with carinal canals, each cortical canal being located near a furrow in the stem surface. This highly characteristic structure allows us to identify fossils related to Equisetum. Figure 21-23. Sporangia of Equisetum occur in groups of 5 to 10 clustered together on a sporangiophore. Sporangiophores are grouped into strobili. Figure 21-24. Calamites was an obvious relative to living Equisetums, having jointed stems with whorls of leaves or branches at each node. A plant like this would have been a small tree about 10 m tall. The evolutionary line of arthrophytes can be traced back through the trimerophytes to the rhiniophytes. In 2 groups of early arthrophytes, the sphenophyllales and the calamitales, a vascular cambium produced secondary xylem. Presumably secondary phloem was produced also, but the preservation of the fossils is not good enough to be certain. These plants became large trees up to 30 centimeters in diameter and more than 20 m tall. They had true monopodial growth, a main trunk, lateral branches, true leaves, and true roots. The equisetophyte vascular cambium evolved independently from that of the lycophytes, yet the two suffered from the same defect. The fusiform initials could not undergo radiolongitudinal division to produce more fusiform initials. As the wood accumulated and pushed the cambium outward, the arthrophyte fusiform initials finally became too large to function, and secondary growth ceased. Although equisetophytes are an ancient clade with only a few surviving species, the plants are by no means primitive, poorly adapted, or rare. They occur worldwide in moist habitats and grow vigorously enough to be weeds in cultivated areas. Figure 21-25. Although Equisetum lacks many derived features, it is by no means primitive or poorly adapted. Here it is a successful weed, competing well with both a corn crop and its human farmers. Ferns. Aside from the equisetophyte line of evolution out of the trimerophytes, probably two more led to the plants that dominate Earth at present: ferns and woody plants. Early ferns first appeared in the Devonian period and then diversified greatly. Whereas equisetophytes only have 15 species of living plants, the rest of the manilophytes have more than 12,000 species, and almost all of those are what are called leptosporangiate ferns. The other three groups of manilophytes have less than 300 species of living plants altogether, and fewer people will be familiar with them. Figure 21-26. This reconstruction of a fern ancestor, Rhacophyton, shows spirally arranged branch systems that had evolved nearly to the point of being called leaves. Some are fertile with sporangia, and some are sterile. The former evolved into sporophylls, the latter into foliage leaves. Table 21-4. Classification of division Pteridophyta. All species in this group are extinct. This family contains almost all the living species of ferns. Ferns can be found in almost any habitat. Moist, shady forests and lakesides are often considered typical fern habitats, but species of Woodsia and Kylantz occur in dry, hot deserts. Salvinia and Azala grow floating on water, and Ceratopterus lives submerged below water. Other genera contain epiphytes or vines. All ferns are perennial and herbaceous, none is woody, but some do achieve the size of small trees, the tree ferns Angiopteris, Cyathea, Nemodaria, and others. Although called tree ferns, they never have secondary xylem. Figure 21-27. A later fern, Phlebopteris, from the Triassic period. Even though the Triassic occurred from 230 to 190 million years ago, plants had evolved so many modern features that this fossil is not an ancestor to ferns in general, but to a particular family of living ferns, the Metoniaceae. The fern sporophyte consists of a single axis, either a vertical shoot or a horizontal rhizome, that bears both true roots and megaphyllous leaves. The vascular system of the stem is an end-arch siphonostele, a derived trait also present in equisetophytes and seed plants. At each node, a leaf trace diverges from the siphonostele, leaving a small segment of the vascular cylinder as just parenchyma. This region is a leaf gap. A vascular cambium has been reported to occur in one fern, Batricium. Figure 21-28. Many ferns grow in hot, dry climates with little rainfall. If transplanted to habitats often described as typical for ferns, cool, moist, and shady, these would die within days. Azala and Salvinia do not look like ferns at first glance, but the structure of their leaves and sporangia shows they are ferns. Several genera of ferns such as this Cyathea are referred to as tree ferns because they have very long vertical trunks and huge fronds. However, unlike trees, these never have secondary growth and are actually giant herbs. Figure 21-29. Anatomy of fern stems is variable, not only from species to species, but also within a single plant. In this Denstoetia and many other ferns, xylem forms a complete ring in transverse section. Phloem occurs on both the exterior and interior of the xylem in ferns. Phloem interior to xylem is extremely rare in seed plants. In this adiantum, the vascular tissue makes almost a complete cylinder. A leaf gap occurs where vascular tissue moves to the leaves. Leaf gaps are characteristic of ferns, they do not occur in the microphyll line of evolution. Members of the microphyll line of evolution, such as this Lycopodium annotinum, do not have pith. Leaves of ferns may be leathery or delicate, only one cell thick in filmy ferns, but many layers thick in most. They have an upper layer of palisade parenchyma and a lower region of spongy mesophyll. Leaves are small or up to several meters long, but they are almost always compound with a rachis and leaflets. Staghorn fern and bird's nest fern have simple leaves. Fern leaf primordia have a distinct apical cell, unlike leaves of seed plants, and as the primordium grows, it curves inward, producing a tightly coiled young leaf commonly known as a fiddlehead. As the leaf expands, it uncoils and becomes flat. Fern leaves, especially larger ones, usually contain a considerable amount of vascular tissue. Leaf veins typically branch dichotomously. Figure 2130, fern leaves have a petiole and a lamina, the lamina being almost always pinnately compound. The midrib is a rachis, and the leaflets may also be compound. Botany and beyond. Box 21-2 form genera. Although we can be fairly sure that a plant like Cyathophyton was the gametophyte of a plant like Rhynia or Cooksonia, we can probably never know for certain. These fossils are the separate pieces of a life cycle, just as the individual fossils of spores, leaves, fruits, flowers, stems, and roots are the pieces of plants. How do we know which fossil parts came from the same species? As a plant dies, its leaves and flowers usually abscise, and thus, we have numerous individual leaf and flower fossils mixed with the remains of bare stems. It is rare to find, at present or in the past, entire meadows or forests that consist of just a single species. Usually many species grow mixed together. We find many types of fossil leaves mixed with many types of fossil stems, flowers, wood, and other plant parts. If we find a branch with leaves or flowers still attached, that is, if there is an organic connection, we can establish which organs are part of one plant. Until we know which parts were portions of the same species, we must use form genera, which are created for types of isolated organs, tissues, spores, or pollen. If a fossil leaf is found which appears to be distinct from all others, it is named as a new form genus of leaf. As more fossil leaves are found in rocks of similar age, some appear to be related to the first and are assigned to that form genus. Mixed with the leaves may be twigs or large pieces of wood. We may be almost certain that the leaves have been produced by the twigs, based on this correlation of common occurrence at the same time and the same place. However, the leaves cannot be assigned to the form genus of the wood unless they are actually found still connected to a twig. Only when an organic connection has been established can two form genera be combined. For spores and pollen, no organic connection ever occurs, but occasionally, an unopened sporangium or anther is found. Therefore, certain spores or pollen grains can be associated with those sporangia or anthers. Even when two form genera have been shown to be parts of the same species, it is still often much simpler to continue using the names of the form genera. In some species, for example Blechnum spicant and cinnamon fern, certain leaves serve only for photosynthesis, and others serve only as sporophylls. However, in most ferns, each leaf does both. On the underside of the leaf are sori, clusters of sporangia where meiosis occurs. Most ferns are homosporous, only two groups of water ferns are heterosporous. Figure 2131 fern leaves have a highly characteristic development. Young leaves are tightly coiled and uncurl as they expand. This is true not only of the rachis, but of the leaflets as well. This is circinate vernation. While young like this, fiddleheads are edible and are cooked as a vegetable. Once mature, fern leaves are inedible. Figure 2132. This cinnamon fern contains both foliage leaves that never bear sporangia and highly distinct sporophylls that do bear sporangia. Most ferns have only one type of leaf. The broad, thin shape of its leaflets is selectively advantageous for photosynthesis, and they bear clusters of sporangia on their underside. These sori are naked, they have no covering. The sporangia of this maidenhair fern occur near the margin of the leaflets and are protected by a flap of tissue called an indusium. Figure 2133. A sorus contains many sporangia, each of which consists of a stalk and the actual sporangium body where meiosis occurs. The sporangium opens when specialized cells of the annulus dehydrate, shrink, and suddenly crack open, expanding rapidly and throwing the spores. Figure 2134. The life cycle of a fern. Details are given in the text. Fern gametophytes, several with the first leaves of young sporophytes. When fern spores germinate, they grow into small, simple heart-shaped or ribbon-shaped photosynthetic gametophytes with unicellular rhizoids on the lower surface but with no vascular tissue and no epidermis. Each usually bears both antheridia and archegonia. Antheridia develop early and can be difficult to detect because they occur among rhizoids. Archegonia develop later, close to the gametophyte apex. When the environment is sufficiently moist, antheridia release motile sperm cells that could easily be mistaken for unicellular green algae except for the absence of plastids. These swim to the archegonia and fertilize the egg. As in equisetum, a fern zygote is retained by the gametophyte that nourishes it. However, the young sporophyte soon produces its first leaves and roots and becomes independent. Its continued growth eventually destroys the small gametophyte. Figure 2135 Development of Eusporangia. Surface cells undergo divisions and the inner daughter cells become sporogynous. Further divisions result in a large number of spores and a thick sporangium wall. Figure 2136 Development of leptosporangia. And the sporangium is initiated by division in just a single surface cell. After a few cell divisions, the sporangium has a stalk, a thin wall, and only a few spores. Eusporangia and leptosporangia. Ferns contain two types of sporangia that differ in fundamental aspects of their development. The eusporangium is initiated when several surface cells undergo periclinal divisions, resulting in a small multilayered plate of cells. The outer cells develop into the sporangium wall, and the inner cells proliferate into sporogynous tissue. This results in a relatively large sporangium with many spores. This is the fundamental type of sporangium, and it exists in virtually all plants other than leptosporangiate. Even the anthers and ovules of flowering plants develop this way. Leptosporangia are initiated when a single surface cell divides periclinally and forms a small outward protrusion. This undergoes several more divisions, which result in a small set of sporogynous cells and a thin covering of sterile cells. Only a few spores are produced. Having leptosporangia is a derived trait occurring only in the clade Leptosporangiidae. Psilotum and Tmesipteris. These two small genera contain the simplest of all living vascular plants. Psilotum is constructed very much like Rhynia and Aglaophyton, and until the mid-1970s, they were often placed together, even though Rhynia and Aglaophyton are extinct. Plants of Psilotum are now known to be highly derived, highly simplified ferns. They are small plants with prostrate rhizomes and upright stems that branch dichotomously. They have an epidermis, cortex, and a simple vascular cylinder with no pith, a protostele. Xylem consists of annularly or helically thickened tracheids. Psilotum is unique among living vascular plants in that it has lost the capacity to make roots and leaves. Shoots do have occasional small, scale-like projections of tissues resembling innations. Figure 2137 The growth habit of Psilotum stems, dichotomous axes with eusporangia. Figure 2138 Vascular structure of Psilotum is simple, a protostele and phloem surrounding the xylem. Protoxylem is exarch, which is similar to the zosterophyllophytes rather than the rhiniophytes. Sporangia of Psilotum are actually considered to be three sporangia fused together. Each is at the end of an extremely short branch, basically a three-branched shoot subtended by a scale. Particularly unusual are the gametophytes, short, branched cylinders less than 2 mm in diameter. Their surface is covered with elongate cells, rhizoids, that anchor and perhaps also absorb. Gametophytes have no chlorophyll but instead are heterotrophic, forming either a symbiotic or a parasitic association with soil fungi. Fungi invade most cells of the gametophyte and provide it with sugars and minerals. Psilotum is the only species whose gametophytes contain vascular tissue. There is a small amount of vascular tissue similar to that of sporophytes. A central mass of tracheids is surrounded by phloem and an endodermis. Psilotum occurs in tropical and subtropical regions. In the United States, it can be found in the Gulf Coast states from Florida to Texas, as well as in Hawaii. Thmacipterus is limited to Australasia, primarily Australia and other South Pacific islands. The term vascular cryptogams. Traditionally, the plants of this chapter have been referred to informally as vascular cryptogams. This name indicates that they have vascular tissue and that because they lack seeds their reproduction is hidden. Although these are two important features, they are shared ancestral features, symplegiorophies, and thus do not indicate they are closely related. What is most striking about these plants are the many features they lack: seeds, flowers, fruits, and so on. However, we construct cladograms on the basis of shared derived features, synapomorphies, not on a lack of features, and consequently, we do not group vascular cryptogams together formally. Also, if we tried to consider vascular cryptogams as a clade made up of several subclades, we would be creating a group that contains an ancestral group but not all its descendants, the ancestor of the monolophyte line is also the ancestor of lignophytes. To leave lignophytes out would be to create a paraphyletic group, which is not correct. Instead, when we use a term such as vascular cryptogam, we are referring to a grade, to basically a level of evolutionary advancement. Similarly, the term bryophyte is often used for all the non-vascular plants, not to indicate that they are related, but that they are at a level of evolution that does not include the presence of vascular tissue. Figure 2139. Gametophytes of Psilotum are small cylinders that live heterotrophically, nourished by endophytic soil fungi. Each gametophyte bears both Antheridia and Archegonia, and because the egg and zygote are retained in the archegonium, the young sporophyte initially is nourished by the parent gametophyte. During early development, there is proliferation of gametophyte tissue as a protective calyptra. As the sporophyte grows and enlarges, it forms an abscission zone and detaches from the gametophyte, becoming independent. At the next level. 1. Zosterophyllophytes and lycophytes. It is difficult to imagine, but the plants that surround us now are in some respects the second flora of Earth. After true plants diverged from algae, the zosterophyllophytes and their descendants the lycophytes dominated Earth and formed extensive forests. This chapter gives only the basics of their biology, and most of what we know about them is their anatomy, some details of their reproduction, and almost nothing of their physiology. Unfortunately, there is no book on zosterophyllophyte biology, but there are review papers and summaries. If you choose these plants for further study, be certain to look up information about the climate at that time. The atmosphere had much more carbon dioxide and it was much warmer than now. 2. The fern life cycle. All plants have a life cycle in which diploid sporophytes alternate with haploid gametophytes. But in most plant groups, this just seems to be something to be memorized, something abstract. It never seems real. But with ferns, we can easily cultivate both generations of the life cycle and have an experience in which we truly understand it. For this independent study, find a fern with sporangia on the underside of its leaves. Collect spores by simply laying the leaf on a sheet of paper overnight. If there are no spores on the paper after several hours, the leaf might be too old and you will need to use a younger one. Sprinkle the spores onto moist potting soil in a plastic flower pot. Cover with plastic wrap, then place the pot in bright, indirect light. Keep it moist by placing it in a deep dish of tap water once a week or so. After 2 or 3 weeks, the potting soil should turn green with gametophytes, just thread-like at first, then heart-shaped after another 2 or 3 weeks. They often grow so thickly that the entire surface of the potting soil becomes covered with gametophytes. After they have grown to the heart-shaped stage, take off the plastic wrap, pour water onto the gametophytes, then cover again and wait. You have just helped them have sex, and sperm cells should be swimming from Antheridia to Archegonia. In a week or 2, the embryos will produce their first leaves. After another month or so you can transplant some sporophytes into individual flower pots. You will have encountered all stages the plant life cycle firsthand. Summary. 1. Some early gametophytes were the most elaborate ever, and subsequent gametophyte evolution from them has involved simplification and reduction. 2. Lycophytes and manilophytes are embryophytes, not algae, because of their multicellular, jacketed sporangia, and gametangia. They have vascular tissue, and thus, they are not mosses, liverworts, or hornworts. Although some have small endosporial megagametophytes, none is retained in the sporangium, therefore, none is a seed. 3. The earliest vascular plants are rhiniophytes, plants with simple dichotomously branched stems, no leaves, and end-arch protoxylem. 4. Zosterophyllophytes were similar to rhiniophytes but had exarch protoxylem and lateral sporangia that opened across their tops. Zosterophyllophytes probably were the ancestors to lycophytes. 5. 3 types of leaf occur in the plant kingdom: gametophyte leaves of non-vascular plants, microphylls of lycophytes, and megaphylls of all non-lycophyte vascular plants. Microphylls evolved from annations, megaphylls are modified telomes, branch systems. 6. Vascular cambia evolved at least 3 times. Cells in the 2 that evolved in ancient lycophytes and equicetophytes could not divide with a radiolongitudinal wall. 7. Trimerophytes were transitional between early vascular plants and euphylophytes. Evolutionary advances in the trimerophytes were pseudomonopodial branching and the first steps in megaphyll evolution. 8. Equicetophytes contain only one small genus of living, herbaceous species and several extinct genera of large, complex, tree-like plants. All had a characteristic jointed structure and various systems of canals. 9. Ferns have many living species and tremendous diversity. No ferns have secondary growth or seeds, but all have megaphylls. Review questions. 1. The earliest fossils that definitely were vascular land plants belong to a genus of extinct plants. They branched, both branches being of equal size and vigor. 2. Draw a plant of Rhynia. Be certain to include the reproductive parts. Now assume that it has a life cycle with an alternation of isomorphic generations and draw a complete life cycle. 3. If Rhynia or its contemporaries were the ancestors to the ferns, how did the gametophytes and sporophytes change during evolution? 4. In the vascular bundles of flowering plants, protoxylem is closest to the center of the stem, and metaxylem is farther out. Is this an end-arch or an exarch arrangement? Seed plants always have just this one arrangement, but what about the early vascular plants? Were they end-arch or exarch, or did both types occur originally? 5. The vascular bundles of flowering plants surround a pith, but the earliest vascular plants had no pith. A vascular system with a solid mass of xylem with no pith is called a stele that does have a pith is called a 6. Rocks that contain fossils of Rhynia also often contain fossils of Cyatophyton. We know that Rhynia was a sporophyte and that Cyatophyton was a gametophyte. Because they grew together, we suspect that they may be the 2 generations of a single species. If so, did Rhynia have an alternation of isomorphic or heteromorphic generations? Also, if this is true, evolution has caused one generation to become more complex, the other to become less complex. Which is which? Explain. 7. There are 2 alternative hypotheses about the life cycle of the early vascular plants, the transformation hypothesis and the interpolation hypothesis. If Rhynia and Cyatophyton are the 2 generations of one species, which hypothesis would be favored? Briefly describe the other hypothesis. 8. What are the zosterophyllophytes? How did they differ from rhiniophytes? Why do we think they are related to lycophytes but not to ferns and seed plants? 9. The reconstruction of Asteroxylon in Figure 2110 shows thin, leaf-like flaps of tissue on the plant's surface. What are these called? Did they ever have stomata in any of the zosterophyllophytes? Did they have vascular bundles? Did they ever become large? 10. What are microphylls? Are they related to the Anaceans of Asteroxylon? 11. The lycophytes once contained many species of large trees that formed extensive forests. Briefly describe plants of Lepidodendron and Sigillaria. Also describe their wood. 12. Name 2 genera of living lycophytes. What are their common names? About how big do they get? Are they leafy or do they have naked stems? In a plant identification book, they would probably be listed with ferns. 13. Trimerophytes were plants that probably evolved from rhiniophytes but with more derived features. In one feature, certain stems grow longer than others, and thus, rather than having dichotomous branching, they have branching. Simultaneously, the positioning of branches became more and. 14. Describe the evolution of megaphylls. What are telomes? 15. Describe the trimerophytes. From what group did they evolve, and what lines of evolution did they produce? Even though all rhiniophytes and trimerophytes are now extinct, would you consider them unsuccessful? 16. What are the 2 common names of equisetums? What is the appearance of the plants? Their approximate height? Look at figures 21-21 and 21-24. Equisetums have strobili and canals. Briefly describe the canals and strobili. 17. Ferns first appeared in the Devonian period. Look at the inside of the back cover. How long ago was the Devonian period? Unlike all the groups mentioned so far in this chapter, most of this group is still extant, not extinct. About how many species of modern ferns are there? 18. Name 2 genera of ferns that are found in deserts. Name 2 that grow floating on water. One that lives underwater. There are ferns called tree ferns. Do tree ferns have woody trunks with secondary xylem? 19. A very important feature of ferns is shown in figure 21-29. Their shoot xylem is not solid as it was in the rhiniophytes. In the evolution of ferns and seed plants, there was the evolution of pith. A stele with a pith is called a. Another significant feature of ferns is that the phloem occurs both to the outer side as well as the inner side of the xylem. Where does it occur in the vascular bundles of flowering plants? 20. You may have noticed that the undersides of fern leaves have brown dots or brown streaks or brown patches. The brown dots are called clusters of where meiosis occurs. If a leaf carries these structures, the leaf is called a. 21. When fern spores germinate, they grow into small, simple heart-shaped or ribbon-shaped with unicellular on the lower surface but with no vascular tissue and no. 22. Briefly describe eusporangia and leptosporangia. Which is the type that occurs in all other vascular plants? Which is the type that occurs in most ferns? Design credits: Hummingbird, Tongo 58 Moment Getty, Green Plant Cells, Shutterstock Incorporated Natalia Ora, Purple Tulip, Shutterstock Incorporated Marie C. Fields, Dandelion, Shutterstock Incorporated Daniel Kreisel, Poppy, Shutterstock Incorporated Saryuri, Plant Icon, Shutterstock Incorporated Vector, Digging Man Icon, Shutterstock Incorporated Z-Art. Chapter 22. Seed Plants I, Seed Plants Without Flowers. Chapter Opener Image. At this stage, these cones of a pine tree have been pollinated and seeds are developing inside, but they are not quite mature enough to be released. These cones are the equivalent of unripe fruits in flowering plants. When these cones were pollinated, they were much smaller, about 1 centimeter long, and the scales were separated from each other so that wind-borne pollen could just settle into the ovules between the cone scales. When these cones are mature, they will be dry, tough, and inedible, like the fruits of many angiosperms. Fir cones fall apart when they are mature, and the seeds simply fall to the ground. In other conifers, the cones expand and drop their seeds, and in still others they open only when heated by a fire. It is very rare for any conifer cone to be fleshy and edible, but those of junipers are. Outline. Concepts. Division Progymnospermophyta, progymnosperms. Aneurophytales. Archaeopterodales. Evolution of seeds. Division Pteridospermophyta, seed ferns. Division Coniferophyta, conifers. Division Cycadophyta, cycads. Division Cycadioidophyta, cycadioids. Division Ginkgophyta, maidenhair tree. Division Gnatophyta. Learning objectives. After reading this chapter, students will be able to explain the process that led to the evolution of seeds. Describe the features of progymnosperms. Name 2 orders in the division Progymnospermophyta. Identify 3 features of seed ferns. Recall 4 features of conifers. Compare seed cones to pollen cones. Explain how cycads differ from seed ferns. List 5 features of the division Ginkgophyta. Name the 3 plant groups in the division Gnatophyta. Did you know? Most of the plants in this chapter are conifers, Christmas trees and their relatives. All species in this chapter have 2 types of cones, pollen cones and seed cones. Before the evolution of seeds, animals had only leaves, stems, and roots to eat, all of which are much lower in nutrients than seeds. The plants in this chapter are usually called gymnosperms, but recent evidence indicates these plants are not one complete clade. Concepts. The life cycle of vascular cryptogams is an alternation of independent, heteromorphic generations. A disadvantage of this life cycle is that the new sporophyte, while developing from the zygote, is temporarily dependent on a tiny gametophyte for its start in life. Consequently, many new sporophytes perish. All of the genes of each gametophyte and half the genes of each new sporophyte embryo are identical to those of the maternal sporophyte, and thus, any mutation that improves the survival of the gametophytes or embryos contributes to the reproductive success of the maternal sporophyte. It would be advantageous if the embryo could use the photosynthetic and absorptive capacity of the leaves and roots of the previous sporophyte. In order for this to happen, the megagametophyte and embryo must be retained inside the maternal sporophyte. This is accomplished by retaining the megaspore and allowing the megagametophyte to develop within the sporangium, figures 22:1. Such an arrangement requires some alteration in the microgametophyte as well because retention of the megagametophyte changes its position. Free-living gametophytes develop on the soil, whereas retained ones develop high on the plant in strobili, a position that cannot be reached by swimming sperm cells produced by microgametophytes living at the soil surface. The modifications that overcame this problem are simple. The megasporophylls with the megagametophytes were arranged in upright cone-like structures, allowing microspores to be carried by wind and dropped into the megasporangiate cone. There they would germinate into a microgametophyte, produce their antheridia and sperm cells, and carry out fertilization. Fertilization itself has changed little, with a moist sporophyll replacing moist soil. These changes produced the first seed plants, the division Pteridospermophyta or seed ferns, and the early members of division Coniferophyta, known as class Cordytales. These 2 groups are now extinct, but their descendants still exist as the living seed plants. In several lines of evolution, those that produced the cycads and ginkgo, this method of fertilization still exists, and beautiful swimming sperms are produced. In other lines, those leading to conifers and to flowering plants, sperms have become non-motile and are carried to the egg by growth of the microgametophyte as a pollen tube. Evolution of seeds was preceded by evolution of a vascular cambium. Vascular cambia had arisen independently in some lycophytes and equisetophytes, but cells in this new cambium could undergo radial-longitudinal divisions, thus allowing the cambium to grow in circumference as wood accumulated. In contrast to the rather slender lycophyte and equisetophyte trees, this new type of cambium could, and still does, produce trees as massive as giant redwoods, oaks, and hickories. We believe that this cambium arose just once, in one group of plants that then gave rise to a monophyletic group of woody plants, the lignophytes. Shortly afterward, seeds originated, establishing the seed plants, spermatophytes. The plants that existed after the origin of wood but before that of seeds were trees that reproduced with spores, the way ferns reproduce. Figure 22.1 and plants that release megaspores. Only limited amounts of nutrient can be placed in a spore, and after spore release, the sporophyte can do nothing more to nourish or protect the gametophytes or new sporophytes. And plants that retain megaspores. Mutations that cause a reduction in the number of megaspores and their retention in the sporangium can be selectively advantageous. The sporophyte can nourish and protect its gametophytes. And also the subsequent progeny sporophytes, at least for a while. The embryo is shown embedded in tissue of the megagametophyte. This occurs in gymnosperms, seed plants that are not flowering plants. In flowering plants, the tiny megagametophyte is quickly replaced by endosperm. Megasporangia that are packed together in a cone composed of sporophylls automatically have a means of catching and retaining microspores. These germinate into microgametophytes with antheridia that release sperms within a millimeter or two of the megagametophyte and its archegonia. Figure 22.1 recently proposed cladogram of seed plants. We are still not certain of the exact evolutionary relationships of all groups of extant seed plants and their extinct ancestors. Notice that in Figure 22.2, many groups emerge from the same point of the cladogram. This is an unresolved polychotomy, and it just means that we do not have enough information to know the order in which the various groups and their innovations arose. One problem is that we cannot extract and analyze DNA from such long-dead fossils. As you read the following descriptions, keep in mind that certain features are often associated with certain others. There appear to be two major suites of characters. Some plants produce a small amount of very soft, spongy, parenchymatous wood, have large, compound leaves and radially symmetrical seeds. This suite occurs in cycads and their relatives. The second suite consists of hard, strong wood with little parenchyma, small, simple leaves and flattened seeds. These occur in conifers and their relatives. The coniferophyte syndrome probably evolved out of the cycadophyte syndrome, but as Figure 22.2 shows, we are not yet certain of this. An old classification from the 1800s grouped all the seed plants together in a single division, Spermatophyta, with two classes, class Gymnospermi and class Angiospermi. The gymnosperms are those plants with naked ovules, that is, ovules located on flat sporophylls, for example, pine cones. Angiosperms are the flowering plants, those with carpels, which are believed to be sporophylls that form a tube-like, closed structure. Fruits are mature carpels. This classification emphasized both the close relationship of all seed plants and the idea that angiosperms originated from some group of gymnosperms, but if angiosperms evolved from some type of gymnosperm, then the group gymnosperms is not natural because it leaves out some of the descendants of the ancestors. In cladistic terms, an incomplete group is a paraphyletic group. Consequently, class Gymnospermi should be abandoned. The divisions of living seed plants commonly accepted now and used in this book are division Cycadophyta, division Coniferophyta, division Ginkgophyta, division Gnatophyta, and division Magnoliophyta. Numerous extinct groups exist, the most significant for us being progymnosperms and seed ferns. Figure 22.3 The vascular cambium of progymnosperms was able to produce a virtually unlimited amount of both secondary xylem and secondary phloem, as in this Calixylon brownii. And progymnosperm wood had large vertical tracheids with pitted walls and an abundance of rays with ray tracheids. Ray tracheids do not occur in flowering plants but are common in conifers as horizontal tracheids that permit rapid conduction horizontally in the sapwood. C. Arianum. C. Newberii. Division Progymnospermophyta, progymnosperms. Trimerophytes were an important group of plants that evolved from rhiniophytes. One evolutionary trend initiated in trimerophytes was pseudomonopodial branching and the first steps in the evolution of megaphylls. These evolutionary trends were continued in horsetails and ferns, which evolved out of trimerophytes. A third group to evolve from trimerophytes was the now extinct progymnosperms. So named because some gave rise later to conifers, cycads, and the other gymnosperms. Like ferns and horsetails, progymnosperms also developed megaphyllous leaves. However, another feature was just as significant, the evolution of a vascular cambium with unlimited growth potential and capable of producing both secondary xylem and secondary phloem. As early as 360 million years ago, the vascular cambium that evolved in progymnosperms was capable of undergoing radiolongitudinal divisions, and thus, it could function indefinitely, producing large amounts of both secondary xylem and phloem. The wood was almost indistinguishable from that of many living conifers. It contained elongate tracheids, most with circular bordered pits. It had little or no axial parenchyma. Rays were tall and uniseriate and consisted of procumbent ray tracheids. This wood must have been strong, effective at conduction, and capable of supporting a large mass of leaves and branches. At least some species, Triloboscilon and Proteocolon, for example, had a cork cambium that produced bark. Progymnosperms produced true woody trees. Trunks were up to 1.5 m in diameter and 12 m tall. Table 22:1 Classification of division Progymnospermophyta. All species of this group are extinct. Figure 22:4 Reconstruction of Archaeopteris, a small tree about 6 m tall. Although extinct now, Archaeopteris flourished about 360 million years ago across northern North America, Canada, and Europe. Although progymnosperm wood was similar to that of conifers, the two groups must be kept separate because progymnosperms did not have seeds or even ovule precursors. The species that arose later were heterosporous, but megaspores were shed through longitudinal slits in the sporangia. They were not retained in an indehiscent sporangium as occurs in seed plants. Although leaves and wood of progymnosperms were quite advanced, their reproduction was remarkably simple. Anurophytales. The order Anurophytales contains the more relictual progymnosperms, such as Anurophyton, Protopteridium, Proteocolon, Tetraxylopterus, Triloboscilon, and Eospermatopterus. They varied in stature from shrubs to large trees, up to 12 m tall. They all had a vascular cambium and secondary growth, but the primary xylem of their stems was a protostele like that of rhiniophytes and trimerophytes. Anurophytes further resembled trimerophytes in having little webbing between their ultimate branches. These could not yet be called leaves. Figure 22:5 Eospermatopterus was a member of the Anurophytales. It had a trunk and what appear to be frond-like leaves, but these were just branch systems. Planation and webbing had not yet evolved. A portion of the leaf of Anurophyton. Archaeopteridales. A more derived progymnosperm was Archaeopteris in the order Archaeopteridales. These were trees up to 8.4 m tall with abundant wood and secondary phloem. Stems of Archaeopteris had a siphonostele, pith surrounded by a ring of primary xylem bundles, much like modern conifers and dicots. Although the fronds of Archaeopterids resembled fern leaves, close examination reveals that they were actually plannated branch systems and that the ultimate leaflets were really spirally arranged simple leaves. Webbing was only partial in A. macilenta, but it is complete in A. halliana and A. obtusa. These can be considered full-fledged megaphylls. Reproduction in Archaeopterids was heterosporous. Megaspores measured up to 300 mm in diameter and microspores only 30 mm in diameter, and each type was produced in its own distinctive sporangium, broad megasporangia and slender microsporangia. Sporangia were terminal on short branches mixed with sterile, leaf-like branch systems. Megaspores were released from the sporangia, not retained. Seeds were not produced. Evolution of seeds. Investigating the life cycle of extinct plants based on fossil material is difficult, but not impossible. In free-sporing species, spores can be identified with sporophytes if some spores were trapped in a sporangium attached to leaves or wood during fossilization. Unfortunately, spores cannot be identified with gametophytes except when the gametophyte is microscopic and develops within the spore wall. If megaspores are retained in the megasporangium, at least some of the investigation is easier. Figure 22.6 During the existence of the plants classified in the extinct genus Archaeopteris, the final transitions from telomes to leaves occurred. Archaeopteris physalis. A. macilenta. A. halliana. A. obtusa. What appear to be veins in the leaves were actually the shoot axes of the telomes. Currently, the earliest known progymnosperm species with heterospory is Cholleria from the Middle Devonian period, approximately 390 million years ago. In an Upper Devonian fossil, Archaeosperma arnoldii, the megasporangium produced only one megaspore mother cell, and this produced only one large, viable megaspore and three small, aborted cells. The megasporangium was surrounded by a layer of tissue, an integument, that projected upward. There was a large micropyle, a hole in the integument that permitted the sperm cells to swim to the egg after the megaspore had developed into a megagametophyte and had produced eggs. This is similar to angiosperm ovules, and some fossils help us understand the early stages. In Gymnosperma kidstonii, the megasporangium was closely surrounded by sterile telomes. In G. latens, the telomes were fused at the base, and in Eurostoma angulari, they were similar to Archaeosperma, fused into one structure except at the tip. The sheath of sterile branches must have been important in trapping wind-blown microspores, but at first it would have allowed them to settle anywhere on the megasporangium. As they fused to each other and to the megasporangium, the space at the top of the megasporangium became the place where microspores settled, acting as a pollen chamber or holding area. As megasporangia evolved into ovules with integuments, other telomes on nearby branches became modified into cupules, which may have later given rise to the carpel in angiosperms. Figure 22.7 A reconstruction of the megasporangium and adjacent telomes of Archaeosperma arnoldii. Each sporangium contained only one large megaspore, the other three products of the spore mother cell meiosis apparently degenerated early. Tissue immediately around the megaspore was the megasporangium wall. Attached to it and extending upward as finger-like projections was a layer called an integument. Above the sporangium was a space surrounded by integument tissue, a calm, wind-free pollen chamber where pollen or spores settled. Around each megasporangium and integument was another set of partially fused telomes. Figure 22.8 A hypothetical evolution of an integument from telomes. In, sporangia are neither clustered or particularly associated with sterile telomes. One megasporangium is surrounded by sterile telomes. The sterile telomes have fused at their bases. Actual fossils that correspond to the hypothesis. Gymnosperma kidstonii. G. latens. Eurostoma angulari. Stamnostoma hiddenans. Simultaneously, microspores were evolving into pollen grains. Changes occurred in the nature of their wall, in their method of germination, and in the nature of the microgametophyte they produced. By the Carboniferous period, just after the Upper Devonian period, four types of pollen had become common. In some, internal structure is preserved well enough to see that these grains already had internal microgametophytes remarkably similar to those of modern gymnosperms. Division Pteridospermophyta, seed ferns. Progymnosperms gave rise to another line of gymnospermous plants in addition to the conifers, the cycadophytes. These are classified as three divisions: Pteridospermophyta, Cycadophyta, and Cycadioidophyta. Figure 22.9 Reconstruction of a swamp forest during the Carboniferous period. The large trees are related to Lepidodendron, and the smaller fern-like plants are seed ferns. Not true ferns. Note the seeds at the ends of some fronds. The earliest seed ferns appeared in the Upper Devonian period, others appeared later. Not all are closely related to each other, they form a grade rather than a clade. Seed ferns were any woody plant with fern-like foliage that bore seeds instead of sori on its leaves. Many resembled modern tree ferns, others were vines. Pteridosperms are thought to have evolved from the Anurophytales because the earliest seed ferns, such as Stenomyelin, had a three-ribbed protostele as in the Anurophytes Triloboxylon and Anurophyton. In later species of Stenomyelin, Calamopodes, and Lygenopterus, most central cells of the stem differentiated as parenchyma, not tracheids. They had a ring of vascular bundles surrounding a pith, an arrangement that also occurs in the stems of all gymnosperms and angiosperms. Seed ferns had a long-lived vascular cambium that produced both xylem and phloem. This too is similar to gymnosperms and angiosperms. Although their wood was basically similar to that of their progymnosperm ancestors, there were interesting differences. Tracheids were much longer and wide enough that several rows of circular bordered pits could occur on each radial wall. Conifer tracheids are so narrow that only one or two rows can fit on each radial wall. Rays in pteridosperm wood were many cells wide, not just one cell wide, and they were very tall, being large wedges of parenchyma. Seed fern wood was manoxylic, much softer and less dense than wood of conifers and progymnosperms, and manoxylic wood also occurs in cycads and cycadioids. Around the stem of pteridosperms was a thick cortex that contained distinctive radial plates of sclerenchyma just below the epidermis. The inner cortex contained secretory ducts. In older plants, a cork cambium and bark formed exterior to the secondary phloem. The cortex was shed with the first bark. Figure 22:10 transverse section of Schopfiastrum stem, showing the beginning of a pith, parenchymatous wood, and the distinctive cortex with plates of sclerenchyma. Leaves of seed ferns were similar to those of true ferns in overall organization, large, compound, and planar. Unlike ferns, however, the foliage leaves of seed ferns bore seeds. Within the seed fern ovule, the megasporangium was large, and bundles of vascular tissue ran into and through it. The integument was attached to the megasporangium only at the base and was vascularized. Seeds could be extremely large, up to 11 cm long and 6 cm in diameter in the now extinct Pachytesta incrissata. Figure 22:11 seed ferns, such as this Emplectopterus, bore seeds along their leaves, not in cones. Otherwise, seed fern leaves were remarkably analogous to the leaves of true ferns in terms of venation and tissue structure. Division Coniferophyta, conifers. Because conifers are familiar plants, they will be discussed first. They are diverse, and all are trees of moderate to gigantic size. The giant redwoods of California reach 90 m in height and 10 m in diameter. Conifers are never vines, herbs, or annuals, and they never have bulbs or rhizomes. Conifer leaves are always simple needles or scales. Leaves of most conifers are perennial, persisting for many years. Leaves of Agathis and Araucaria remain even on very old trunks. The venation of conifer leaves is often simple, with just one or two long veins running down the center of a needle-shaped leaf or several parallel veins and scale-shaped leaves. Leaf veins have an endodermis and a tissue called transfusion tissue, consisting of transfusion parenchyma cells and transfusion tracheids. The latter are more or less cuboidal and have prominent circular bordered pits. Transfusion parenchyma is intermixed with the tracheids, the two cell types forming a complex three-dimensional pattern that facilitates transfer of materials between the ordinary vascular tissues and the mesophyll tissue outside the endodermis. Table 22:2 Classification of division Coniferophyta. All species of this group are extinct. Figure 22:12 Division Coniferophyta is large and diverse, containing many familiar plants. Pine, European larch, big tree redwood, western juniper, Figure 2213 conifer leaves are never compound. They are always simple and leathery, needle-like in some, and scale-like in others. In bristlecone fir, each leaf has two white bands where stomata are located. Scale-like leaves of incense cedar. Pines have two types of leaves, small, papery brown scales on the long shoots and the long needles borne by the short shoots located in the axils of the papery scale leaves. The long needles were produced last year. Just as in their progymnosperm ancestor Archaeopteris, wood of modern conifers lacks vessels, and their phloem lacks sieve tubes. Tracheids are so narrow that only one or two rows of circular bordered pits occur on their radial walls. All conifers have pollen cones and seed cones, most of which are woody, but in Juniperus and Podocarpus, seed cones superficially resemble the fruits of flowering plants. The conifers are still a very successful group, forming extensive forests covering over 17 million kilometers. In many of these forests, flowering plants exist only as herbs, shrubs, or small trees growing in the conifer's shade. Figure 2214. The vascular bundle of this Douglas fir leaf has an endodermis, xylem, phloem, and transfusion tissue. High magnification of a pine leaf bundle showing secondary phloem and transfusion tracheids. Casparian strips are visible in the endodermis walls. The pines are good representatives for closer examination. The trees are monopodial, with one main trunk bearing many branches. The wood is composed exclusively of tracheids, but annual rings, spring wood, and summer wood are all visible because large diameter tracheids are produced in the spring, followed by narrow diameter tracheids in the summer. Rays are thin and tall and contain both ray parenchyma and ray tracheids. Resin canals, which produce the thick, sticky pitch, run vertically among the tracheids and horizontally in the rays. The wood has almost no axial parenchyma. Phloem contains sieve cells and albuminous cells and also has tall, narrow rays. A cork cambium produces a thick, tough bark that provides excellent protection even from forest fires. Pines, like several other conifers, have two types of shoot, each with a characteristic type of leaf. Tiny papery leaves occur on long shoots and in their axils are short shoots that produce the familiar long needle leaves. The leaves have many xeromorphic characters, thick cuticle, sunken stomata, cylindrical shape. Like all conifers, pines have both pollen cones and seed cones. Pollen cones are simple cones with a single short unbranched axis that bears microsporophylls. Microspore mother cells undergo meiosis and form microspores, then each of these develops endosporially into a small gametophyte with 4 cells, one of which is a generative cell as in flowering plants. The gametophytes are shed from the tree as pollen and carried by wind, a small percentage land in seed cones, but the great majority land elsewhere and die. Figure 2215. Wood of pine, like that of all conifers, lacks vessels. It consists almost exclusively of tracheids. Growth rings are visible because late wood contains narrow, thick-walled tracheids, whereas early wood contains wide, thin-walled tracheids. Pine tracheids are narrow and their circular bordered pits are wide, and thus, only one row of pits fits on a given wall, unlike the dozens of small pits that cover each wall of an angiosperm vessel. Seed cones are more complex than pollen cones. They are compound cones, each consisting of a shoot with axillary buds. The short axis bears leaves called cone bracts rather than sporophylls. Each bract has an axillary bud that bears the megasporophylls. In some fossil conifers, the individual structures can still be seen, but in all modern conifers, extensive fusion has occurred. The axillary bud is microscopic, and its megasporophylls are fused laterally, forming an ovuliferous scale. In larch, fir, and Douglas fir, the ovuliferous scale is still distinct and can be seen, but in most conifers, it is fused to the bract. Figure 2216. Phloem of conifers contains sieve cells, not sieve tube members. Sieve cells are long and narrow with sieve areas over much of their surface, but they never have horizontal cross walls with enlarged sieve pores. The sieve areas in this pine are particularly abundant and easy to see. Figure 2217 in Cedrus atlantica, the nature of short shoots is obvious because they form more needles each year and so slowly grow into a visible shoot. Figure 2218 pollen cones typically occur in clusters near the ends of branches. As the microsporangia dehisce, pollen is liberated to the wind and blown away for distribution. Such a method of gene transfer is inefficient because so few pollen grains land on seed cones. It is successful primarily because conifers grow as dense forests, where each conifer is surrounded by hundreds of potential partners. Pollen cones are simple cones. They have one single stem axis and bear microsporophylls. These are not microsporangia but rather leaf-like structures that bear microsporangia. Inside each megasporangium, a single large megaspore mother cell undergoes meiosis, with 3 of the resulting cells degenerating and only one surviving as the megaspore. The megasporangium does not dehisce. The megaspore is retained inside and grows into a large cenocytic megagametophyte by free nuclear divisions and may have as many as 7,200 nuclei. It is much larger and more complex than the simple embryo sac of angiosperms. Development can take as long as a year, but finally walls form, converting the coenocyte into a cellular megagametophyte. 2 or 3 archegonia form as sets of cells, each surrounding a large egg. Conifer eggs are gigantic cells loaded with carbohydrate and protein. The egg nucleus, although haploid, is swollen to a volume much larger than that typical for entire cells. It is probably filled with DNA synthetases and RNA polymerases, ready for extremely rapid activity once karyogamy with a sperm nucleus occurs. Figure 2219 and the microspore of pine has 1 cell and 2 large air bladders that increase its buoyancy in air. The microspore develops into a small gametophyte in a process similar to that in pollen of flowering plants, except that a few more body cells are formed. First, 2 mitotic divisions produce 2 small body cells that degenerate and a large cell that divides, resulting in a generative cell and another body cell, called the tube cell. As in angiosperms, the body cell becomes the pollen tube, and the generative cell divides into 2 non-motile sperm cells. Figure 2220. The 2 air bladders of pine pollen are easily visible in this light micrograph. The addition of the hollow bladders adds virtually no weight to the pollen but increases its volume so that the pollen's density is decreased and it does not sink so quickly in air. This pollen grain of pine has germinated and its tube cell is elongating. Figure 2221. The long 3-pointed bracts of this Douglas fir cone are the true leaves of the cone axis. Each one contains an axillary bud whose leaves have fused together side by side into the flat, shield-like ovuliferous scale just behind each bract. These fused leaves that constitute the ovuliferous scale are actually megasporophylls. If an ovuliferous scale were pulled from the cone and turned over, the 2 ovules would be visible. The scale obviously does not look like a set of leaves fused together, but look at figure 22.6. Figure 2222. In seed cones of pine, all parts are fused together, even the bract is fused to the ovuliferous scale. When morphologists first began working on pine cones without knowing about fossil structures, they were completely baffled and could not explain such a complex structure. Larch. Monkey puzzle. Taxus has individual seeds, each surrounded by a red, fleshy aril. There are no ovulate cones in this genus. Figure 2223. This section of a pine cone was made just as the megaspore mother cells were about to begin meiosis. Each ovuliferous scale carries 2 ovules, each of which contains an integument and a nacellus, the actual megasporangium. As in flowering plants, each nacellus usually has only 1 megasporocyte and produces only 1 surviving megaspore. 2.25. Conifer pollen arrives before the egg is mature, and more than a year may pass between pollination and fertilization. A massive pollen tube slowly digests its way toward the megagametophyte as the egg forms. Because the megasporangium does not open, a passageway digested by a pollen tube is necessary. This differs greatly from the free-living gametophytes of mosses and ferns, which have exposed archegonia that sperm swim into. The 2 or 3 eggs in one megagametophyte can all be fertilized, but only one zygote develops into an embryo, the other 1 or 2 die. A zygote does not immediately form an embryo in conifers. Instead, some of the first cells elongate as a suspensor that pushes the other cells deep into the megagametophyte. These other cells, called the proembryo, develop into the embryo. There is no double fertilization as occurs in angiosperms. Rather, the female gametophyte continues to grow and acts as a haploid nutritive tissue similar to endosperm. The mature embryo has the same organization as an angiosperm embryo but always has many cotyledons, not just 1 or 2. The seed also resembles that of a flowering plant, but it is born in a cone, not a fruit. In 2 conifers, the cones become fleshy, fruit-like, and brightly colored, red in Podocarpus, blue in Juniperus. Figure 22-24 ovules of pine and other conifers are much larger than those of flowering plants. This cellular megagametophyte is not as large as a moss or liverwort gametophyte, but it is much more plant-like than the megagametophytes of angiosperms. Above the 2 eggs is the thick, indehiscent megasporangium. Sperms cannot swim to the archegonia and eggs, they must be carried by a pollen tube that digests its way through the megasporangium. Between the megasporangium and the integuments is the pollen chamber. Figure 22-25. Just as in angiosperms, the zygote of conifers produces a suspensor that thrusts the proembryo into the megagametophyte. The suspensor can divide and form multiple embryos from each zygote, but only one embryo survives. A conifer seed looks remarkably like the seed of a flowering plant. It has a seed coat derived from the integument, an embryo with several cotyledons, and a nutritive tissue that is actually the megagametophyte, not endosperm. After fertilization, the parental sporophyte transports large amounts of nutrients, supplying everything needed for the embryos, which grow completely heterotrophically. It also fills all cells of the megagametophyte with carbohydrates, proteins, and mineral nutrients. This acts just like the endosperm of flowering plants and is often called endosperm. Division Cycadophyta, cycads. Modern cycads are frequently confused with either ferns or young palm trees because they have stout trunks with pinnately compound leaves. Most cycads are short plants less than 1 or 2 m tall, but macrozamia can reach heights of 18 m. The trunk is covered with bark and persistent leaf bases that remain on the plant even after the lamina and petiole have abscised. Internally, cycad stems are similar to those of seed ferns. A thick cortex containing secretory ducts surrounds a small amount of manoxylic wood. Tracheids are long and wide, and rays are massive. Even very old stems have only a small amount of wood. Most support is provided by the tough leaf bases. A prominent pith contains secretory canals. Unlike seed ferns, cycad foliage leaves do not bear ovules. Instead, cycads produce seed cones and pollen cones, each on separate plants. Cycads are always dioecious. Pollen cones consist of spirally arranged shield-shaped microsporophylls that bear clusters of microsporangia. Upon germination, pollen grains produce a branched pollen tube and large, multiflagellated sperm cells. Seed cones are variable, with those of Cycas revoluta usually considered the most relictual. In this species, the seed cone is a large, loose aggregation of leaf-like, pinnately compound megasporophylls. 6 to 8 large ovules occur near the base, but the upper half of the megasporophyll is rather leaf-like, similar to the ovule-bearing organs of some seed ferns. Megasporophylls of the cycad Zamia floridana are considered more derived. They are shield-shaped and bear only 2 ovules, and the entire cone is quite compact. Cycad ovules are like those of seed ferns, having a large, vascularized megasporangium and a loosely attached, vascularized integument. Although Cycadophyta was a much larger group with many more species in earlier times, currently, it contains 9 or 10 genera and approximately 100 species. Modern cycads are highly prized ornamentals in the warmest parts of the United States. Only a few can withstand freezing temperatures in winter. They are almost all tropical with an unusual distribution. Some occur in Cuba and Mexico, others in Australia, still others in Southeast Asia or Africa. Cycads are now believed to have been widespread and to have occupied many habitats, but in most areas, they have become extinct. The few remaining, widely scattered species are the survivors of a formerly extensive group. Figure 22-26 Aspects of the vegetative body of cycads. Dione has a short, stout trunk with tough, leathery, pinnately compound leaves. Note the huge seed cone. Leaf veins with transfusion tissue similar to that in conifer leaves. Transfusion tracheids extend from veins into the leaf blade. Stomata are visible, slightly out of focus, in the lower epidermis. Cycads have only small amounts of wood, it is parenchymatous. Figure 22-27 Pollen cone, more than 30 cm long, in Cycas circinalis. It is a simple cone, with one axis that bears microsporophylls. Microsporophylls in cycads may bear many microsporangia. Sperm cells of cycads have hundreds of flagella and must swim to the egg cell. Zamia. Figure 22-28 Cycad seed cones are simple, with megasporophylls borne on the only axis. They have no bracts or axillary buds. Cycad megasporophylls are not fused structures as are ovuliferous scales of conifers. Instead, the megasporophyll is simple and strongly resembles a foliage leaf, at least in Cycas circinalis. It is not difficult to see how this type of sporophyll could evolve from a seed fern sporophyll. Division Cycadioidophyta, cycadioids. The cycadioids had vegetative features almost identical to those of cycads. The two groups differ only in subtle details of the differentiation of stomatal complexes and in leaf trace organization. On such characters alone, cycadioids would never be considered distinct from cycads. However, individual cones of cycadioids contained both microsporophylls and megasporophylls. Each ovule had a stalk, and the megasporangium was surrounded by an integument that extended out into a long micropyle. Between the ovules were thick, fleshy scales. Microsporophylls were located below the cluster of megasporophylls and curved upward, enveloping the megasporophylls. Each microsporophyll was cup-shaped and contained numerous microsporangia. Division Ginkgophyta, maidenhair tree. This division contains a single living species, Ginkgo biloba. It may seem unusual to erect an entire division for a single species, but G. biloba is itself unusual. It looks very much like a large dicot tree with a stout trunk and many branches, but its wood is like that of conifers. It lacks vessels and axial parenchyma. It has broad leaves, but they have dichotomously branched veins like seed ferns, not reticulate venation like dicots. Ginkgos have both short shoots, which bear most of the leaves, and long shoots. Reproduction in ginkgo is dioecious and gymnospermous, but cones are not produced. Instead, ovules occur in pairs at the ends of a short stalk and are completely unprotected at maturity. Pollen is produced in an organ that resembles a catkin, having a stalk and several sporangiophores that each have 2 microsporangia. Like ovules of pteridosperms and cycads, the ovules of ginkgo are large and develop a 3-layered seed coat. Figure 22-29 Vegetatively, cycadioids such as Cycadioidea were similar to cycads. They had a broad cortex and a tough outer protective layer formed by persistent leaf bases. Figure 22-30 A tree of Ginkgo biloba could easily be mistaken for a dicot tree. Ginkgo leaves are broad. They have dichotomous venation like leaves of cycads and seed ferns, not reticulate venation like dicots. Microsporophylls occur in small, cone-like clusters mixed with foliage leaves on short shoots. Ovules occur in pairs at the end of a stalk-like megasporophyll. The ovule itself, the integument, is exposed, no bract or ovuliferous scale protects it. A ginkgo tree itself is beautiful and is a popular ornamental because the leaves turn brilliant yellow in autumn, but the microsporangiate trees are preferred. When the megasporangiate trees produce seeds, the outer fleshy layer of the seed emits butyric acid, which has a putrid odor that is difficult to tolerate. The exact ancestors of ginkgos are not known, but they must have been one of the seed ferns or a closely related group. The cladogram of figure 22.2 proposes that cycads and ginkgos have a common ancestor, but DNA analysis cannot include the seed ferns, all of which are extinct. Ginkgos became abundant during the Mesozoic era, especially in the mid-Jurassic period. Fossilized remains of leaves and wood can be found in almost all areas of the world, especially in high latitudes such as Alaska, Canada, and Siberia near the North Pole, and Patagonia, South Africa, and New Zealand near the South Pole. Ginkgos began to die out early in the Tertiary period, but two species, G. biloba and G. adiantoides, persisted. The latter species became extinct during the Pliocene epoch, approximately 10 to 12 million years ago. Division Gnatophyta. Gnatophyta contains three groups of enigmatic plants, Gnidum with 30 species, Ephedra with about 40 species, and Welwitschia mirabilis, the only species in the genus. Gnidums are mostly vines or small shrubs with broad leaves similar to those of dicots. They are native to Southeast Asia, tropical Africa, and the Amazon basin. Plants of Ephedra are tough shrubs and bushes that are very common in desert regions in northern Mexico and southwestern United States and dry mountains in South America. Their leaves are reduced and scale-like. The few living plants of Welwitschia exist only in deserts of South Africa or in cultivation. They have a short, wide stem and only 2 leaves, but the leaves grow perennially from a basal meristem, becoming increasingly longer. Figure 2231 Gnidum. Plants of Gnidum strongly resemble dicots, having broad leaves and woody stems. Ovules are not borne in cones. Figure 2232 Ephedra. Plants of Ephedra often occur in dry areas and strongly resemble many types of desert-adapted dicots. Although their reproductive structures are gymnospermous, the microsporangiate cone could be mistaken for a staminate imperfect flower. The naked ovules reveal that they are not angiosperms. All 3 genera are unusual in being gymnosperms with vessels in their wood. This had been thought to show that they might be related to primitive angiosperms. However, their vessel elements evolved from tracheids with circular bordered pits, whereas those of angiosperms were derived from scalariform tracheids. Furthermore, angiosperms are thought to have evolved from vessel-less ancestors, the vessels evolving after flowers, not before them. Unlike the pollen cones of all other gymnosperms, those of gnatophytes are compound and contain small bracts. Seed cones are also compound and contain extra layers of tissue around the ovules. The tissue is variously interpreted as an extra integument, bract, or sporophyll. The few fossils of gnatophyte organs or tissues are only several million years old, too recent to be of much help in understanding the evolution and ancestry of the group. The pollen is distinctive, being spindle-shaped and having narrow ridges. It is easy to recognize, and fossil pollen of this type occurs as far back as the late Triassic period, but pollen has not helped reveal their origins either. Figure 22.2 includes them as one of several groups emerging from the same point. Certain aspects of their anatomy and reproduction have been interpreted as indicating that gnatophytes and flowering plants constitute 2 sister clades with a common ancestor. If so, the 2 would form the group Division Anthophyta or Anthophytes. However, there are too few data to be confident of this, and currently, it is best to recall the principle of skepticism, realize that we do not yet know, and must continue searching for more data about their phylogenetic relationships. Remember that Figure 22.2 is a model, a hypothesis. Figure 22.33 Welwitschia mirabilis. Whole plant with torn leaves. Microsporangiate strobili. Megasporangiate strobilus. At the next level. 1. Animals and gymnosperms. Cooperation between animals and angiosperms is widespread. Animals pollinate flowers and disperse fruits. This also occurs with gymnosperms more often than is commonly realized. Some birds cache seeds of conifers in several locations, and then forget about some of them, which are thus able to germinate far from the parent tree. The bright red arils of taxus and the fleshy soft blue cones of junipers attract animals. The flesh-covered seeds of maidenhair tree must have been eaten by some animal that is now extinct. Cycads have such large pollen cones that wind is probably the main pollination vector, but beetles have been found carrying pollen from plant to plant. You may have to gather information bit by bit, but the interactions of animals and gymnosperms is an interesting, underappreciated area. 2. Conifers in the last 100 million years. Giant redwood trees now exist in just a few valleys in California and need all the protection we can give them so they do not go extinct. But several million years ago they were abundant and widespread in extensive forests. Starting about 50 million years ago, Earth became cooler and its climate changed. The redwoods provide a good example of how some species react as conditions change, how refugia are important to the survival of species. The same thing happened to many other species and genera of conifers in the last 100 million years. The ecology of redwoods is well known and worth examining as an introduction to the paleoecology of conifers in general. 3. Cycad sperm cells swim with hundreds of flagella. We don't usually think of plant cells as being good swimmers, but in fact the sperm cells of all cryptogams and several species of seed plants must swim to the egg cell. In fact, the sperm cells of cycads put those of animals to shame. Cycad sperm cells are large and have hundreds of flagella arranged in a long spiral that wraps around half the cell. Just as in animal flagella, plant flagella need a basal body, and these must be produced by the hundreds as each cycad sperm cell matures. The cell biology of cycad sperms is a bit different from most plant cell biology, but is definitely interesting. Summary. 1. In a seed, the embryo draws its nutrition from the surrounding megagametophyte, which in turn is supplied with nutrients by the parental sporophyte. The sporangium and associated structures provide protection for the megaspore, megagametophyte, and embryo. 2. In all modern seed plants, the megasporangium does not open, therefore, a pollen tube must deliver the sperm cells to the archegonia. Sperm cells are still motile in many seed plants. 3. Progymnosperms arose from trimerophytes with the evolution of a vascular cambium of unlimited growth potential which produced solid wood with little parenchyma. The evolution of true seeds also began in the progymnosperms, first with retention of the megaspore and then with formation of an integument. 4. One order of progymnosperms, Archaeopterodales, gave rise to conifers. Another progymnosperm order, Anerophytales, evolved into pteridosperms, cycads, cycadioids, and perhaps angiosperms. 5. Conifers constitute a diverse division of large trees with solid wood, simple pollen cones, and compound seed cones. 6. Pteridosperms gave rise to cycads and cycadioids. Important evolutionary changes were formation of pith and of ovules on more or less unspecialized foliage leaves. Their wood contained abundant axial parenchyma. 7. Cycads are modern seed plants with compound leaves, parenchymatous wood, and simple seed cones consisting of an axis and a rather leaf-like megasporophyll. 8. Ginkgo biloba is the sole living species of its division. It resembles a dicot tree except that its leaves have dichotomous veins and it bears naked ovules, surrounded by neither a cone nor a carpel. 9. Reproduction in neophytes is gymnospermous, but plants of ephedra and needham resemble dicots. Welwitschia has only 2 leaves, which grow indeterminately from basal meristems. Important terms. Angiosperms anthophytes compound cones cone bracts gymnosperms integument lignophytes long shoots manoxylic wood micropyle ovuliferous scale pollen chamber proembryo progymnosperms pycnoxylic wood seed ferns short shoots simple cones spermatophytes suspensor. Review questions. 1. What is the disadvantage of a life cycle in which alternate generations are completely independent of the preceding generation? Are there advantages to this type of life cycle? 2. What is a cone? How are cones of conifers similar to those of lycophytes and cycads? How do they differ? 3. The classification of the seed plants has varied. If a person groups all seed plants together in one large division spermatophyta, then they have 2 classes, the class with naked seeds and the class, plants with carpels. On the other hand, many people use 4 divisions, not 1. List the 3 divisions of gymnosperms, and the 1 division of flowering plants. 4. If angiosperms evolved from some type of gymnosperm, then the group gymnosperms is not natural. Why not? In cladistic terms, an incomplete group is a group. 5. Describe progymnosperms. What were the significant evolutionary advances that characterized progymnosperms? 6. The section Evolution of Seeds describes the fossil seeds Archaeosperma arnoldii. Its megasporangium was surrounded by a layer of tissue, called and that projected upward, and there was a hole that permitted sperm cells to swim to the egg. If you have studied flowering plant reproduction, how many of these same features occur in flowering plants? 7. In progymnosperms, microgametophytes produced and released sperm cells that could swim, but not very far. What is a pollen chamber and how did it help fertilization? 8. Are any conifers herbs? Annuals? Vines? Do all conifers have needles like pines? 9. Describe a conifer pollen cone. Why is it a simple cone? Describe a conifer seed cone. Why is it a compound cone? 10. Seed cones of conifers are more complex than pollen cones. Seed cones are cones, each consisting of a shoot with axillary buds. The main axis of the cone bears leaves called, rather than sporophylls, and each of these has an axillary bud that bears the axillary bud is microscopic, and its are fused laterally, forming an scale. 11. Unlike pollination in flowering plants, conifer pollen arrives before the egg is mature, and more than a may pass between pollination and fertilization. The pollen germinates, and a massive slowly digests its way toward as the egg forms. 12. In most conifers, as the seeds mature, the cone scales and ovuliferous scales become hard and tough. A typical pine cone, however, the cone becomes red and fleshy, and it becomes blue and fleshy. 13. Seed ferns are a group of extinct plants. They resembled tree ferns, but what did they have on their leaves instead of sori? 14. There is a good chance you have seen cycads. What do they look like? What 2 other types of plants can they be confused with? Do cycads ever have simple leaves? 15. Do any seed plants have flagella? Do their sperm cells have flagella? Look at figure 22-27. 16. What is the one and only species in the division Ginkgophyta? Being part of the gymnosperms, does it have needle-shaped or scale-shaped leaves? The leaf venation is unusual because the leaves have dichotomously branched veins not reticulate venation. 17. What are the 3 groups classified in Gnatophyta? 18. Would it have been possible, on a theoretical basis, for an indehiscent megasporangium to evolve before integument-penetrating pollen tubes evolved? Why or why not? What problem would be involved? Design credits: Hummingbird, Tongo 58 Moment Getty, Green Plant Cells, Shutterstock Incorporated Natalia Ora, Purple Tulip, Shutterstock Incorporated Marie C. Fields, Dandelion, Shutterstock Incorporated Daniel Kreisel, Poppy, Shutterstock Incorporated Saryuri, Plant Icon, Shutterstock Incorporated Vector, Digging Man Icon, Shutterstock Incorporated ZArt. Chapter 23. Seed Plants 2, Angiosperms. Chapter Opener Image. The poppy flower shows two important ways in which angiosperms differ from other seed plants. First, the pollen-producing organs and the ovule-producing ones occur together in most flowers, whereas they always occur as separate cones in gymnosperms. Second, carpels are closed, which means they have no opening that allows pollen to just fall into the carpels and onto the ovules. Instead, the pollen must germinate and send a pollen tube through the stigma and style, carrying the two sperm cells downward to the ovule and egg. This results in a prolonged period of contact during which the stigma and style might reject the pollen tube if it is not the proper type. In many angiosperm species, the carpels exert considerable control over which pollen tubes are allowed to grow and which are not. Outline. Concepts. Changing concepts about early angiosperms. Classification of flowering plants. Basal angiosperms. Monocots. Commelinoid, monocots. Eudicots. Basal eudicots. Rosid clade. Asterid clade. Box 23-1 Plants and People, Maintaining Genetic Diversity. Box 23-2 Plants and People, Vegetarianism, Alternatives to Eating Meat. Learning Objectives. After reading this chapter, students will be able to list several derived characters of angiosperms. Explain the changes that occurred in the transition from gymnosperms to angiosperms. Compare monocots and eudicots. Name the several groups that are extant descendants of the basal angiosperm clades. Describe the features of monocots. Give 10 familiar examples of monocots. Identify 3 distinctive features of eudicots. Discuss the evolution of eudicots into basal eudicots, rosids, and asterids. Did you know? There are more species of flowering plants than all other groups of plants combined. Some flowers, such as those of cattails, maples, and oaks, are so tiny and inconspicuous you might not realize they are flowers. The largest flower in the world, Rafflesia arnoldii, is about 1 m in diameter and is produced by a parasitic plant. All angiosperms produce seeds, but orchids produce very tiny, underdeveloped dust-like seeds that are almost microscopic. The embryo is just a few dozen cells or less. Concepts. Of all the clades that have arisen since embryophytes became distinct from charophyte green algae, the flowering plant clade, the angiosperms, contains the greatest number living species, 257,000. These are all classified together in a single division, the Magnoliophyta. It is common to refer to angiosperms as the most advanced group of plants, or the most derived. Occasionally, they are called the peak of plant evolution, but these statements should be examined carefully. Certainly the group as a whole has a greater number of derived features than any other group, and especially important is the large number of derived characters that are unique to angiosperms and that distinguish them from other plant clades. The group is named for having flowers, a significant synapomorphy that at once unites all angiosperms and separates them from all other clades. Other important derived characters are numerous methods of mutually beneficial interactions with animals that result in the plants being pollinated or having their seeds distributed while the animals receive nectar or pollen or some other food. At the same time, angiosperms and animals have become more adept at fighting each other, as angiosperms evolved to provide reward for pollinators, natural selection also was giving them numerous new types of antiherbivore compounds, ranging from outright poisons to milder chemicals that deter feeding. Wood of almost all angiosperms is more complex than that of conifers. It contains vessels, which provide a low-friction pathway of water conduction, and it contains axial wood parenchyma, which makes wood a living tissue that protects itself from wood-boring insects and fungi and can influence the distribution of water as it is pulled upward through the plant. Do these features, however, make angiosperms the most advanced of all groups? Keep in mind that the simple spore-based reproduction of ferns has allowed them to reproduce successfully, spread into many habitats, and diversify into numerous sizes and shapes. Conifers also are extremely successful. One clade in particular, Pinaceae, is a relative newcomer, having arisen within the conifer clade after the angiosperm clade had already made quite a few advancements. Pinaceae is diversifying and speciating rapidly, producing not only many new species but also new genera. The point is that we must not think about evolution as a linear progression forward and upward, with each new group leaving the previous ones behind. Instead, think in terms of real plants and populations. Some population of a species encounters an isolation barrier that causes it to diversify until at some point some of its members cannot reproduce sexually with others, and at that point, two very similar but distinct species exist. Both are free to continue evolving. After the early embryophytes became distinct from charophytes, they diverged into two clades, the zosterophyllophyte lycophyll clade and the rhiniophyte euphyll clade. Both were extremely successful, undergoing large numbers of modifications and diversifying greatly. After the early ferns originated, they too continued to diversify, with most variation occurring in habit, leaf size and shape, type of sporangium, type of indusium, and type of sporophyll. None evolved to have wood, flowers, or fruits, but they are not more primitive than the angiosperms. Instead, different features have evolved in the two groups. The earliest fossils clearly recognizable as parts of flowering plants are preserved pollen grains in rocks more than 130 million years old. We have not found any fossilized leaves or flowers from that time, and thus, we believe the earliest flowering plants must have lived in hilly or drier areas, areas where few sites were suitable for fossilizing plant parts. Large calm lakes with fine-grained sediments are best for fossilizing plants, and these are rare in dry areas or rocky hillsides. The first angiosperms probably were small trees or shrubs because plants of this type survive well in drier regions and because the most likely ancestors, other seed plants such as seed ferns or gymnosperms, were woody trees. As discussed earlier, the term gymnosperm may refer to an unnatural group. It is being used in this chapter to mean all the seed plants except angiosperms. The evolutionary transformation of a gymnosperm into a flowering plant was not a simple process, and it involved many alterations. The most obvious was the conversion of gymnospermous sporophylls into stamens and carpels, resulting in the formation of flowers. Because many gymnosperms have rather flat, leaf-like sporophylls, arranged in spirals around an axis, and microsporophylls never occur with megasporophylls except in cycadioids, those flowering plants that have similar reproductive parts are believed to be the most relictual. For example, in a group known as the basal angiosperms, such as Amborella, Nymphiales, Austrobaleales, and Magnoliales have flat stamens without distinct filament and anther portions, arranged in spirals, and their sporogynous tissues form relatively large, prominent internal masses. In most angiosperm carpels, the edges of sporophyll primordia crowd against each other and grow shut, sometimes leaving a visible suture, sometimes closing so completely that no sign of a seam remains. This is called a closed carpel, and it develops into a fruit that encloses the embryos as they develop into seeds. Carpels in the basal angiosperms are leaf-like, resembling young leaves whose blades have not yet opened. Instead of a stigma and style, the ovary edges have rows of secretory hairs that secrete a thick liquid that both seals the seam and functions as a stigmatic surface. Whereas ovules in gymnosperms are exposed on the megasporophyll surface, the folded nature of the megasporophyll in flowering plants offers ovules some protection, and probably much more important is the fact that the pollen tube must now interact with the stigmatic area in addition to the nacellus, allowing more opportunity for the sporophyte to test the pollen. Weak pollen or that of the wrong species can be inhibited from germinating, and the sporophyte permits only healthy pollen of the proper species to pass through the stigma and reach its ovules. Fewer ovules are wasted by exposure to improper or unhealthy pollen. Figure 23(1). Several groups of angiosperms have stamens with what are believed to be relictual features. These features have not undergone much evolutionary change, so they are still similar to the ancestral features. The stamens of these